In this episode, we are going to be talking about transcription factors. Specifically, we are going to be addressing the different types of transcription factors that there are in the cell. We're going to be talking about how they function, how they act, and how they are regulated. And at the end, we're going to cover two specific examples of transcription factors that are very important for you to be aware of. One of them is p53, and the other one is E2F. So let's get started. So the first idea that is essential about transcription factors is that they are always required for transcription to take place in mammalian cells. So let's put that idea here. Always require. And the reason for this is that in mammalian cells, RNA polymerase is not sufficient. It cannot recognize promoters by itself in the absence of transcription factors. So transcription factors play a really critical role in regulating the whole process of transcription. Now, when it comes down to the different types of transcription factors that we have in the cell, there are two main different types. There are the ones that we call general transcription factors, and there are the so-called specific transcription factors. The general transcription factors are always required, which goes with this, they are always required.
That's me, always required.
Specific transcription factors are only required for a specific subset of genes. So, require only for a specific subset of genes. All right, now the general transcription factors are named after the RNA polymerase that they function for. So we have general transcription factors that work with RNA polymerase 1, so those will be called TF1 factors. There are transcription factors, general transcription factors that work with RNA polymerase 2, so those will be called TF2 because they work with RNA pol 2.
RNA pol 1.
And finally, there are transcription factors that work with RNA polymerase 3, so those will be called TF3. They work with RNA pol 3. So, different types of general transcription factors. Now, when it comes down to the specific transcription factors, which are the ones that we are going to be talking for the most part, when it comes down to the specific transcription factors, we have two big different types of transcription factors. The ones that will promote transcription, those will be the activators, and the ones that will limit transcription, those will be the repressors. All right, now when it comes down to the specific transcription factors, most of them have as a characteristic feature of them the fact that they are made of two different domains, two different functional regions within the protein that are fairly well separated within the primary structure of the protein. One section of these specific transcription factors has the ability to bind to DNA and recognize specific sequences in the DNA, and that's why they are referred to as the DNA binding domain. The other region of the transcription factors is the one that is involved in recruiting other proteins or other factors, and therefore is referred to as either the activator domain or the repressor domain.
Alright, let's make a line here to separate these two different concepts.
Alright, so that's a very characteristic feature of transcription factors, the specific transcription factors, that they have two very well separated regions within the linear structure of the protein, within the primary structure of the protein. And in fact, each one of these two different regions can work to some degree independently of the other. So the DNA binding domain is able to interact with specific sequences of DNA, even if it were to be separate from the activation domain or the repressor domain. Whereas the repressor domain is able to, or activator domain, is able to interact with other proteins, other factors, even in the absence of the DNA binding domain. So they are kind of like functionally independent from each other, and that's why they are referred to be independent functional domains within this protein. So they are independent functional domains. Okay, and again, let me just re-emphasize, this DNA binding domain is the one that recognizes specific sequences in the double-stranded structure of the DNA. All right, so now let's address how they function, how these different specific transcription factors are able to provide their function for the cell. So let's start with the activators. The activators function by either of two different mechanisms. So let's start here with the activators. They either are able to bind directly and recruit either mediator or the general transcription factors. That's one mechanism of action for these activators. The other mechanism of action is by recruiting coactivators. And we're going to go in just a second about what those coactivators can be. All right, so now let's move on with the repressors. How do repressors work? So they work by three basic different mechanisms. The first mechanism is by outcompeting with an activator. So imagine that you have a specific DNA sequence that is recognized by an activator. So you have here a factor that will be activating transcription. This is an activator. If you have another factor that binds to it and prevents the binding of that activator, that other factor that will prevent the binding of the activator could be acting as a repressor. These have the DNA binding domain, and they can then interact with that same DNA sequence and therefore block the binding of an activator. So that's the first mechanism of action for repressors. The second mechanism of action is by blocking the recruitment of mediator or general transcription factors directly.
Recruitment of mediator or general transcription factors.
If you directly block the recruitment of mediator or general transcription factors, you will also be decreasing transcription overall. And the final mechanism by which repressors can actually repress transcription is by recruiting co-repressors, which is opposite to activators. Instead of recruiting coactivators, repressors can recruit co-repressors. All right, so now that we have the basic mechanisms by which these repressors and activators can function, let's then talk about something that is quite important to refer to, and that is what these are, coactivators and co-repressors. So what are they? They are the kind of proteins that have the ability to modify chromatin structure. And there are different types of coactivators and co-repressors. So let's actually go through them. So coactivators, let's start with them. Coactivators are the ones that produce a more open type of chromatin, which is then easier to be accessible by the general transcription factors, therefore making chromatin more accessible, easier to transcribe. So coactivators include histone acetyltransferases, or HATs. So let's spell it out. And coactivators also include some histone methyltransferases. Some, not all. There are a very specific subset of histone methyltransferases that will act by opening chromatin as well. And a good example of a histone methyltransferase that is a coactivator is the one that methylates histone 3 in lysine 4 and makes a trimethylation event in that lysine. All right, so co-repressors, on the other hand, are the kind of factors that will lead to a more close chromatin structure. Let's put that here to make the difference. So these promote an open chromatin structure, whereas the co-repressors will promote a closed chromatin structure. And within the co-repressors, we have the histone deacetylases, or HDACs. So they remove acetyl groups. HATs add acetyl groups to histones. Histone deacetylases remove acetyl groups from histones. So HDACs are histone deacetylases. Within the core repressors, we also have some histone methyltransferases, but these are the type that promote a more close chromatin structure. So let's put those down here. Now I'm gonna go on my knees, almost as if I was begging for forgiveness for making this confusing diagram. So I'm going on my knees. Histone methyltransferases. So they are the same, they are called the same way, histone. But in this particular case, these histone methyltransferases are the ones that promote a more close chromatin structure. And there are two examples that I think are relevant for you to remember. Those are the ones that do histone H3 lysine 9 trimethylation event, as well as the ones that do histone H3 in lysine 27 a trimethylation event there. So these are two examples of histone methyltransferases that are inhibitory. And finally, the last type of modification that leads to a closer, more inaccessible chromatin structure is methylation of DNA. So DNA methyltransferases, which typically methylate DNA sequence in cytosines that are followed by guanine residues. So CPG motifs. All right, so all of these, when they are recruited to DNA, lead to a more close chromatin structure. All right, so let's talk now about how you regulate the activity of transcription factors. So transcription factors are regulated by three different mechanisms. The first level of regulation for transcription factor is by shortening the half-life of the protein. And this is a feature of most transcription factors. Most transcription factors have a very short half-life, so they are rapidly degraded. So one way to regulate the activity of a transcription factor can be to stabilize, so that the half-life of the protein is increased, allowing the transcription factor to then accumulate in the cell. So they are rapidly degraded, but they can be stabilized. Now, the rapid degradation of most transcription factors typically occurs by a polyubiquitination-dependent mechanism. So rapid degradation involves polyubiquitination, followed by proteosomal degradation. So that's the first mechanism to regulate the activity of specific transcription factors. The second mechanism to regulate the activity of specific transcription factors is by regulating the cellular localization of these factors. Now, transcription factors, to be active, they need to be localized in the nucleus, because that's where DNA is located. So that's where transcription actually takes place. So for a transcription factor to be fully active, it has to be able to translocate from the cytoplasm into the nucleus across the nuclear pore complex. So it requires nuclear localization. And this idea implies that transcription factors must have a nuclear localization sequence. And that nuclear localization sequence will allow transcription factors to then interact with the proteins that typically act as cofactors to facilitate import into a nucleus, and that is the importins. So part of the regulation of the activity of transcription factors is to regulate their nuclear localization. They can be either exported out of the nucleus, because some of them have nuclear export signals. So during specific conditions, the interaction with nuclear export factors might actually be facilitated, whereas the interaction with importins may actually be blocked. The final level of regulation, and it is one that actually affects the other two different levels of regulation, is post-translational modifications. And I'm going to add to that / interactions with other proteins, or protein-protein interactions. So, when these transcription factors are modified by certain types of post-translational modifications, you may actually affect either the half-life of the transcription factor, either stabilizing it or destabilizing the transcription factor, or you may also facilitate the nuclear localization or the cytoplasmic localization of the transcription factor. So post-translational modifications, as well as interactions with different proteins, with other different types of proteins, can actually affect either of the other two mechanisms that I had already mentioned for regulating the activity of the transcription factors. And these actually lead us to talk about the two specific examples that I want to address to close this video, and that is p53 and E2F. So before we do that, let's erase the board and then move on to talk about p53 and E2F. So let's talk now about p53 and E2F. And let's start with p53. Now, p53 is the kind of transcription factor that is regulated primarily by affecting the stability of p53. So p53 is produced at kind of basal levels in the cell. However, very soon after the protein is produced, p53 is degraded by a polyubiquitination dependent mechanism. So there is a specific ubiquitin ligase that targets p53 for polyubiquitination. So you get the attachment of numerous ubiquitin molecules. Ubiquitin is a protein, remember that. So many different ubiquitins are attached to p53, and the attachment of those multiple ubiquitin molecules to p53 then targets p53 to be degraded by the proteasome. However, when you have DNA damage in the cell, so you have DNA, and there is a very specific type of DNA damage that is detected by a specific type of kinase, and this kind of DNA damage is double-stranded breaks. So remember, you have double-stranded DNA breaks. That is a signal that activates the activity of a kinase, a protein kinase, that is able to recognize that signal. That protein kinase is known as ATM. So ATM recognizes that type of DNA damage and becomes activated. And when ATM is active, it is able to then go on and phosphorylate another kinase. This kinase is called the check kinase. The check kinase, specifically one that is known as check2, is then able to phosphorylate p53. So this one is a kinase, and in its kinase activity, it has the ability to phosphorylate p53. And this is a really critical event in the life of p53, because once p53 is phosphorylated, so now we have p53 in the phosphorylated form, p53 in this form cannot be recognized by the ubiquitin ligase, so it will not be polyubiquitinated. In the absence of polyubiquitination, p53 will become stabilized, and the concentration of p53 will increase within the cell. So when the concentration of p53 increases, then p53 has the ability to translocate into a nucleus and start affecting the transcription of very specific sets of genes. The first set of genes that are targeted by p53 include a gene that codes for a protein known as p21. And p21 is a protein that will help prevent the activation of CDK2 and cyclin E. So p21 is produced, it is transcribed, the protein is translated, and once the protein is made, p21 interacts with CDK2 and cyclin E, and this interaction prevents the activation of CDK2 cyclin E. It remains inactive, and in the inactive form, this prevents progression into S phase, therefore producing arrest in G1. So the cell ends up being arrested in G1. Now, if the signal continues, if the signal that is triggering the phosphorylation of p53 continues, meaning the DNA damage has not been repaired, it remains in place, check kinase will continue to be phosphorylated by ATM, and in that form, the check kinase will continue to phosphorylate p53, and therefore p53 will continue to increase its concentration inside the cell. So now you will have even larger concentrations of p53 within the cell, and now that you have more p53 within the cell, now p53 will be able to bind to other promoters for which it has slightly lower affinity for them. So now p53 is able to not only hit certain promoters such as p21, but also the promoters of other genes, including the genes for puma and noxa. So the promoter regions for these other genes, so this is the promoter for a gene known as puma. p53 will now activate that promoter, not only the promoter for p21, but also a promoter for puma. And puma is one of the pro-apoptotic BCL2 family members. Pro-apoptotic member of the BCL2 family of proteins. So now that puma is being synthesized, it will actually promote the cell entering into apoptosis. So important, again, the essential idea here is that p53 is normally rapidly polyubiquitinated and therefore degraded by a proteasome-dependent mechanism, but when the cell is undergoing double-stranded DNA breaks, therefore a type of DNA damage, there are a set of kinases that include ATM and the check kinases that become activated, and the check kinases as well as ATM have the ability to phosphorylate p53. And when p53 is phosphorylated, it becomes stabilized because it cannot be polyubiquitinated when it is in the phosphorylated form. Therefore, now the concentration of p53 can start to augment within the cell, and when there is enough p53, it will hit the promoters for which it has high affinity, starting with p21. But if the DNA damage continues, then the concentration of p53 within the cell will continue to increase, and now p53 will be able to start hitting other promoters, including the promoters of pro-apoptotic genes, including puma and noxa. So that's p53, a very specific example of a transcription factor that is regulated mostly by post-translational modifications, ubiquitination and phosphorylation. All right, and now let's talk about the other transcription factor that I wanted to deal with in this video, and that is E2F. So E2F is another specific transcription factor that plays an essential role within the cell. So E2F is normally produced at fairly high levels within the cell, and when E2F is produced, it has the ability to bind to a specific set of promoters within the cell. So it binds to promoters that include the genes that code for DNA polymerase alpha, proliferating cerebronuclear antigen, or PCNA, and it also has the ability to bind to a promoter for cyclin E. So E2F will bind to those different promoters, and it will remain bound to those promoters. However, the moment that it binds to those promoters, it typically becomes associated to another protein, a protein known as RB. And when E2F is bound to those promoters in the presence of RB, E2F acts as a repressor. So it blocks the transcription of all of those genes. So transcription of those 3 different types of genes is blocked. So under these conditions, when E2F is binding to RB, it acts as a repressor. However, when RB becomes phosphorylated, so if RB becomes phosphorylated, then RB becomes dissociated from E2F. So now E2F is free of RB, and when E2F is free of RB, now it continues to interact with those same promoters that it was interacting before, but now that interaction is the type that will be activating transcription. So now the genes will be transcribed, and therefore these different proteins will be produced, and the synthesis of these 3 different types of proteins, DNA polymerase alpha, pCNA, cyclin D, will allow the cell to go into S phase. So the phosphorylation of RB is one of those essential steps that allow the cell to progress past the restriction point and into this type of events that will lead the cell into S phase. So when cyclin E starts accumulating, then cyclin E will interact with CDK2, and that will be an interaction that will be critical in driving the cell into S phase. So E2F is one of those transcription factors that have the ability to act as a repressor when it is interacting with RB, or as an activator. And it acts as an activator when it is free of the interaction with RB. So this is a really important example to keep in mind, because it gives you a sense of how specific transcription factors can actually be able to have dual functions as repressor or activators, depending upon what kind of proteins they are interacting with. The interaction with RB allows E2F to act as a repressor. In the absence of RB, E2F acts as an activator. Well, I hope that this gives you some broad perspective on the way that transcription factors act, specifically how specific transcription factors actually are able to regulate transcription in the cell, and how this will lead to the activation of the transcription of different sets of genes within the cell. And as exemplified here, you can either be activating genes that will hold progression through a cell cycle, or genes that will actually facilitate progression through the cell cycle, or even the transcription of genes that will lead the cell to go into apoptosis. I hope that you find this video informative and concise and to the point enough, and if you actually do like it, as always, give it thumbs up and subscribe to my channel. Thanks so much.